This is Team Smellsfishy1 from Postville, Iowa Plant the Moon Challenge, team number 25140. We wanted to find out if lunar soil would produce usable food resources. When colonizing the moon, astronauts will need a source of protein that they can get by raising fish. The cultivation of fish, such as tilapia, provides a rapid and efficient source of animal protein. The fish will produce emulsions, which will need to be disposed of. These emulsions and dirty fish water can be used to significantly boost plant growth. Let's move to our key points. We hypothesized when using fish waste and hydrobeads we would be able to turn the lunar regolith into a hospitable place for plant growth. A unique part of this experiment was using the PVC pipe and sponge design pot as an instant plant watering system. We used cellulose sponges to separate the Y connector from the main pipe and then added on the pipe which would house the seedlings. The sponge easily kept the nutrients and soil separate from the water, ensuring the plants wouldn't drown nor dry out. When watering, we used two different types of water, distilled water and water from the classroom fish tanks. We also used two different types of aqua beads, beads hydrated with distilled water and beads hydrated with water from the fish tailings. Fish tailings are the fish feces and extra fish food that collects at the bottom of the fish tanks. With the help of Iowa DNR, we were able to collect some from the Chuck Gipp Fish Hatchery in Decorah, Iowa. The fish tailings would allow for extra fertilizer to add to the nutrients already in the soil. Onto soil combinations. Moon control was the controlled lunar experiment with just the lunar regolith. To all other lunar samples, we added 15% earth soil to provide bacteria and other soil microbes. In two of the three other lunar experiments, we added 15% fish tailings and 20% hydrobeads. We had three other experimental control groups using earth soil. Earth Control 3 was 100% earth soil. Earth Control 1 was 80%, and 20% distilled water hydrobeads. And Earth Soil Control 2 was 80% earth soil and 20% fish water aquabeads. We then planted the baby spinach, which had germinated using the paper towel method, into our growth pots. We measured the progress every week on Thursdays, weather permitting. On the first Thursday after planting, we clipped off the cotyledons to ensure the plant's energy would go towards its true leaf growth. This experiment was monitored every day and the PVC pipes were getting filled with the needed water amounts. Multiple tubes connected to a fish tank bubbler, as seen in the photos, were also placed inside the main pipe to ensure that the plants were getting a healthy amount of oxygen to their roots. After 3 weeks, we had noticed that some of the experiments were condensing due to the cellulose sponges decomposing. By week 4, they'd completely collapsed. This harmed plant life in Experiment 2 and led us to replacing the sponges in Experiment 2 and Earth 3. The photo shows the complete collection of all of the sponges, including artificial sponge replacements after the growth session. At the end of our experiment, we saw great growths in all but the complete lunar regolith. What surprised us is that it wasn't a complete Earth control group, which was the greatest leader in mass and height documentations. Instead, it was Experiment 2 with a combination of 50% moon regolith, 15% earth soil, 15% fish feces, and 20% fish-infused hydrobeads. It was also watered using the fish water from the classroom tanks, suggesting that the addition of fish-based organic matter and the use of hydrobeads will enhance plant growth and increase the yield of edible baby spinach leaves. Despite plant loss from the sponge deterioration, this experiment was a definite success, suggesting that aquaculture waste products and unique watering techniques could play a key role in agricultural setups in space. This has been Smellsfishy1, team number 25140, planting baby spinach.